Hi, and welcome everybody to the front row. My name is Jamie Williamson, and I am the Executive Vice President here at Scripps, and we are kicking off our front row for 2022. Today, we are going to hear from Professor Xiang-Lei Yang, and she is a professor in the Department of Molecular Medicine. And her talk is going to be, it's kind of a remarkable talk and a remarkable evolution of a long program. I mean, Xiang-Lei started out her career as a structural biologist looking at the structures of fundamental components of what we call the translation apparatus. So expression of the genetic code goes from DNA into protein in the process of translation. And we tend to think of this as very conserved. It's present in all cells, and it's the same. And what's really remarkable about her work is that she has found ways in which certain of the components of this apparatus have been specialized to get extra functions. And those functions are actually traceable to disease. And she's been able to use this to come up with some exciting new strategies to treat some very specific kinds of diseases. So I'm going to let her give you more background about that and about herself. So without further ado, Xiang-Lei, please share your screen and take it away. And I'll pop back in at the end and we'll have a chat.
Well, welcome to the front row lecture. I'm Xiang-Lei Yang. And Jamie, thank you for the introduction. And let me also give a brief introduction about myself. I grew up in southern China in a city called Changsha. My parents decided that I should be a scientist before I was even born. I was not a particularly good student, and I did not like to be told of what I should become. So as a way to escape, I came to the United States and become a student, a graduate student studying biophysics. I know that sounds a bit ironic, but that was the only way I could escape. As a graduate student in Andrew Wang's lab at the University of Illinois at Champaign, I learned how to determine atomic structures of DNA and proteins. Although they are considered large molecules, but they're still so tiny that we cannot see them with some special technologies. And as a graduate student, I learned some of those technologies, such as X-ray crystallography and NMR spectroscopy. Those skills helped me landed a postdoc position in Paul Schimmel's lab at Scripps Research. In the Schimmel lab, I started by working on determining the structure of a family of protein called tRNA synthetases, and become fascinated by the enigmatic biology behind them, and also intrigued by their connections to human diseases. Eventually, I was fortunate to become a faculty member at Scripps Research and to have my own lab to join force with the Schimmel lab to tackle this problem together. So here I am, honored and excited to have this opportunity to tell you about our beloved tRNA synthetases and our journey to understand them and how the understanding can be exploited for treating human diseases. Are you ready? I want to start my lecture by introducing the central dogma of biology. As you know, DNA is the blueprint of life. It contains all the information needed to create an organism. The blueprint is stored in the safe box in the headquarter of a cell called the nucleus. The process of taking the information from the DNA into making protein is what we called the central dogma of biology. I know dogma has a negative connotation, but here it really just means the central principle of biology. Making protein is super important. A large percentage of our body weight is made of proteins. Proteins have very important functions. In order to talk to you right now, I need a protein called myosin to move the muscles in my mouth. When we breathe in fresh air, we need a protein called hemoglobin to bring the oxygen from the lung to the rest of our body. The antibodies that we heard so much about during the last couple of years are proteins as well. The process of making protein starts from the DNA and copying the DNA or a piece of the DNA into the mRNA in this process called transcription. Only the mRNA is allowed to leave the nucleus and go into the cytoplasm to do the protein synthesis function. And this step of taking the information from the mRNA to make protein is called translation. Let me just say here that the main character of today's talk, the tRNA synthetases, are involved in translation. It is called translation because it is literally like translating one language into another. The language of DNA and mRNA are in nucleotides of 4 different kinds: A, T, C, G for DNA, or A, U, C, G for mRNA. Whereas the language of protein are made of 20 different amino acids. The rule to translate the nucleotide language into the protein language is called the genetic code. This chart is the genetic code. It tells you that every 3 nucleotides, 1, 2, 3, encode for 1 amino acid. For example, AUG encodes for methionine, CUC, CUU, CUA, CUG, all codes for the same amino acid, leucine. Based on these rules, the mRNA can be translated into protein. Remarkably, the genetic code is universal. All lives, from bacterial in our gut to yeast that we use to make bread, everything in our backyard or the zoo, from plants to animal, and ourselves, all use the same genetic code, the same rule to translate genes into proteins. This tells us that all lives share the same common ancestor, and the genetic code was established very early during evolution. What is responsible for establishing the genetic code? It is a family of protein called amino acid tRNA synthetases. How was the amino acid tRNA synthetase involved in establishing the genetic code? Well, first of all, it needs a partner, tRNA. tRNA is RNA. So it uses the same nucleotide language, and it has a 3 nucleotide anticodon that can directly read the gene. So tRNA is also called the decoder. But that's not enough. You also need the synthetase to recognize the right tRNA from a large pool of different tRNA molecules, and the right amino acid, again, from a large pool of different amino acids, and put them together like this. So through this matchmaking function of the tRNA synthetase, the relationship between the 3 nucleotide anticodon or codon and the amino acid are established early on in evolution. And this rule is followed in all modern organisms whenever a protein is made. Each of the 20 protein-making amino acids have its own tRNA synthetases. For example, leucine has the corresponding tRNA synthetase, leucyl tRNA synthetase, or LuRS. Serine corresponding to SARS, glycine corresponding to GLARS, histine corresponding to HISARS, and etc. This is a very important slide of my lecture today, so I hope I can make it clear to you. Let me tell you the point first, then I'll explain. The point is tRNA synthetases are very special in their ability to bridge our nature with our nurture or our environment. What is our nature? Our nature is our DNA. our gene, and its products such as proteins. All we have is human genes and human proteins. We don't have a cow protein or a soybean protein in our body, even though we drink cow milk and eat tofu. They just get digested right away to provide us with the amino acid building block for us to make our own protein. What is our nurture? It's our food, our environment, it's the nutrient, it's the oxygen that we need for survival and for living a good, healthy life. Different organisms may take the same food, breathe the same air, but they're not self. They're not intrinsic, they're extrinsic. tRNA synthetase are special because they have this ability to be directly connected to our nature through this decoding and protein synthesis function I talked about just earlier. And at the same time, tRNA synthetase also have a direct relationship with our nurture. It needs the amino acid and the energy in the form of ATP that we ultimately get from our food to do the job of expressing our nature. So tRNA synthetase are at this very unique position, bridging the nature with the nurture, the self with the non-self, the intrinsic with the extrinsic. This bridging position of the tRNA synthetase make them uniquely fit in the job to coordinate and balance those two worlds. The balance and coordination is what we call homeostasis. Let me recap with this simple picture. tRNA synthetase are at the crossroad between our nature and our nurture. They have the ability to respond to the changing environment and connecting the change to the expression of our gene, and thus can help to maintain our body's homeostasis. Let me give you a simple but elegant example from recent work of scientists in South Korea to illustrate on this point. Under the situation where there's a lot of sugars or glucose available, which is the most important source of energy for all organisms, LURS does its normal job, binding to leucine, put leucine onto the cognate tRNA for protein synthesis. However, when there's a limited supply of glucose, for example, when we're starved, LURS get modified and cannot bind the amino acid anymore. So instead, being used for protein synthesis, now the amino acid is used to produce some energy through the mitochondria to help with the starving situation. So LURS is sensitive to the environment, can adapt to the environment to find a balance between making protein and producing energy for the benefit of the organism. In order to better understand how tRNA synthetases regulate this balance or homeostasis, we took inspiration from evolution. And here is the thinking. As evolution goes on, over billions of years, life evolved from simple organisms such as E. coli or yeast into complex organisms with sophisticated functional systems. As a result of evolution, we need more and more communications and coordination between different systems, different cells, and between the self and the environment, the nature and the nurture, as we talked about. And let's take a look to see how tRNA synthetases are changed during evolution. The most obvious place to look is the tRNA synthetase gene itself. We could see that they become longer with extra sequences that can fold into unique structural domains. The appearance of the new domain correlates with the increasing complexities of the organism, suggesting the new domains mediates new functions that are important for complex organism. For the rest of my lecture, I would like to share 3 stories of how we understand the role of tRNA synthetase in maintaining the homeostasis of our body, and how the understanding can be exploited for treating human diseases. The first story is about serine tRNA synthetase, or SRS, and our discovery of a function of this protein in the headquarter of the cell in regulating blood vessel growth, and how our finding leads to a potential therapy for treating cancer. The story starts from zebrafish. 3 independent labs from MIT, Kyoto University, and UCSF found mutations in SRS cause defective vasculature. And the defect is the overgrowth of blood vessels. A former postdoc in my lab, Yishi, or Eric, who is now heading his own lab at Nankai University in China, immediately recognized that from fish to humans, all vertebrate SRS have some extra sequence at the end that coincide with the presence of an elaborated vasculature network called the closed circulatory system. After he took a closer look, he found a nuclear localization signal sequence, or NLS, that is embedded in this extra sequence, which would direct a protein to enter the headquarter. As I mentioned, the headquarter is the nucleus, where the DNA is stored. Proteins are made in the cytosol. If the protein made contains an NLS, the protein can be directed to the headquarter. To do the decoding job, SRS do not need to go to the headquarter. So why SRS goes to the headquarter, and does this have anything to do with developing blood vessels? It turns out that SRS goes to the headquarter to control transcription, the gene copying process. The gene that SRS controls is a key regulator for blood vessel development called VEGF-A. SRS binds to the VEGF-A gene and blocks it from being copied. However, there's another protein called c-MYC, which has the opposite activity. It also binds to the VEGF-A gene, but activates its transcription. In order to develop a normal and functional vasculature, the opposite function of MYC and SRS must be balanced with each other. It's like yin and yang. However, in fish with defective SRS, the balance goes to the MYC side, and more VEGF-A genes are copied, more blood vessels are developed, and give the blood vessel overgrowth phenotype that you saw earlier. So nuclear SRS is involved in this balancing act by acting as a negative regulator of blood vessel growth. Then we ask ourselves, if SRS is a negative regulator of blood vessel growth, under what circumstances the function must be inhibited? We know that under low oxygen condition, which is also called hypoxia, more blood vessels needs to grow so that more oxygen can be brought to the site. This is when we think the function of SRS should be inhibited. A well-known factor, which is a key driver for this process, is called HIF. And the discovery of HIF and how HIF is activated under hypoxia was awarded with the 2019 Nobel Prize in Physiology or Medicine. This was a key piece of data made by Eric that reviewed what was going on with SRS during hypoxia. Under hypoxia, the two positive regulators, HIF and c-MYC, bind to the VEGF-A gene, are increased as we would expect for activating new blood vessel growth. What's interesting is that SRS binding to the VEGF gene is decreased during hypoxia. And there is an inverse relationship between the increase and the decrease, suggesting that removal of SRS from DNA may be required for the MYC and HIF to bind. If that is the case, we may have a way to prevent MYC and HIF binding to the VEGF-A gene. And that would be very useful in treating cancer. And that way can be trying to do something to make the SRS to stick to the DNA. So of course, in order to manipulate the SRS binding, we have to understand what happened to cells during hypoxia. Eric found out when cells are short of oxygen, cells are chemically modified by phosphate groups, which are negatively charged. And this negative charge will repulse cells from the DNA, which is also negatively charged. So when cells is not bound to the DNA because of this repulsion, now HIF and MYC can bind to the DNA and turn on the VEGF expression and blood vessel growth. Both HIF and MYC are cancer-causing genes, and their role in activating blood vessel growth is well known. But what was not known before the study is that their activity is dependent on the inactivation of cells. And as said, if we can figure out a way to keep cells bind to the DNA, even under low oxygen condition, we may have a therapy for cancer. Well, one way to do that is to substitute the modification site from serine into alanine, something like alanine, which cannot be modified. Then the cells with this alanine substitution can stick to the DNA even when cells are under the low oxygen condition. In this case, even HIF and MYC are around, they cannot bind to the DNA to be functional. So let me further explain this idea in the context of cancer. In tumor, due to fast growth of tumor cells, a low oxygen environment is very common. In that situation, HIF is activated, MYC is overexpressed, SARS is inactivated by phosphorylation to allow more blood vessel to grow to support the cancer growth. But if we can introduce this sticky form of SARS, we might be able to inhibit tumor growth. And that is indeed the case. Human triple-negative breast cancer cells, which are super aggressive, were injected into mice and they grow into large tumors. However, if we introduce the sticky form of SARS, SARS-AA, into the cancer cell, the tumor growths are strongly inhibited. This shows the potential for SARS in a modified form to be used for anti-cancer therapy. Okay, in this example I just showed you, first of all, SARS has developed a new function to enhance its ability to control the expression of our gene, which is our nature. Here, SARS not only decodes our gene to make protein, but also controls what proteins to make or not to make. And this control of gene expression plays a balancing role in blood vessel growth. In addition, SARS is sensitive to the environment, which is the nurture, and in this case is oxygen. This sensitivity allows the control of SARS on gene expression to be further connected to the environment. This example also shows that by studying homeostatic regulation of tRNA synthetase, we may come up with insight and opportunities that can be exploited for treating diseases. Also, as shown by this example, the homeostatic regulation of a tRNA synthetase can be hidden. This hidden roles may only become visible when our normal homeostatic process is disrupted, such as in the mutant fish, or in a state of human disease. So next, I'm going to tell you about diseases that are linked to tRNA synthetases. There are two main diseases. One is a genetic disease called Charcot-Marie-Tooth disease, or CMT in short. CMT is the most common inherited neurological disorder. It was named after three physicians who first characterized the disease 130 years ago. And still, there's still no treatment available these days for the disease. This is a disease of the peripheral nerve, which are nerves that are outside our brain and the spinal cord. The disease mostly affects long peripheral nerves that goes from our spinal cord to our body extremities. The nerve degeneration leads to muscle weakness and sensory loss, at our body extremities, such as hands and feet, legs, and arms. CMT patients have difficulties in walking, in balancing, and in using their hands. They're physically handicapped. With the permission of their mother, I can show you this picture of Amy and Ashley, twin sisters whose CMT are caused by mutations in glycol-tRNA synthetase, or GLARS. They lost the ability to walk, and you can see from this picture their muscle wasting in the leg and in the hands. It turns out that tRNA synthetase is the largest gene family linked to CMT. There are a total of 6 different tRNA synthetase whose mutation cause CMT. And there's no need for you to pay attention to the details here. I just want to show you that most of the CMT-linked tRNA synthetase have a new domain called the web domain that was added to the synthetase at some point of evolution. To study a disease, either for understanding the disease mechanism or for developing therapeutics, it is so important to have animal models of the disease available. We're very fortunate to have both mouse and fly models that captures the hallmarks of the CMT disease. Here is a mouse model of the CMT on the right. The CMT mouse is, in this case, is also caused by mutation in glycol-tRNA synthetase, just like in the girls that you saw in the previous picture. Compare with the healthy mouse on the left, that the CMT mouse that you can see, it walks with the dragged limbs and the body touching the ground. This is because that their limb muscles are so weak that can barely support their body weight. Our entry point to understand the disease mechanism was to study the structure of the CMT-linked tRNA synthetases and to understand what is the difference between the normal and the mutated tRNA synthetase. Structure is the look. How the protein looks like can tell us about its function. Like we say, form follows function. Wei Wei He, a former graduate student in my lab, discovered that CMT mutation induced structure opening in GLARS. And then David Brockwell, a former postdoc, realized that the structure opening effect is a common phenomenon for CMT causing mutations in other tRNA synthetase as well. The point is, this structure opening effect creates new surfaces that enable new interactions with the mutant tRNA synthetase. Over the years, many people in our lab and other labs in the world have identified many things that went wrong as a result of the aberrant structure of the CMT mutant and the new interactions they made. This including deficiencies in stimulating nerves from the outside and in transporting materials along the long nerve. And there are also problems in the headquarter as well as in the space where proteins are made, all of which are important for maintaining the health of a long peripheral nerve. This multitude of problems making targeting a specific pathway for curing the disease potentially difficult. However, we now know 2 ways that the CMT disease can be fully rescued in the mouse model. Work from Eric Stockebone's lab in Europe have shown that overexpressing the corresponding tRNA partner of the tRNA synthetase that is mutated can fully rescue the CMT. And scientists in our lab, Ge Bai and Yao Tong, found that removal of the new domain from GLARS can also fully rescue the CMT. And Ge is now also heading his own lab in Zhejiang University. Although we still don't fully understand how those manipulations rescued the disease, but we can think about it from the homeostatic regulation point of view. As I explained earlier, tRNA synthetases can be sensitive to the environment and can respond to the environment by regulating gene expression to help maintain a balanced homeostatic state. In CMT, the mutations in tRNA synthetases can cause an aberrant response to the environment. In this case, the specific stress factors are unknown, but they can be a variety of internal or external forces. The open structure of the mutant tRNA synthetase and the resulting new interactions may lead to over-response to the environment and the stress factors. Because we think the new domains enhance the regulation in response to the environment, as I showed in the previous example of SARS, removal of the new domain in the tRNA synthetase may be viewed as a way to suppress this dysregulation to restore the balanced state. On the other hand, although adding more tRNA partners under this dysregulated state of CMT may be a way to achieve the opposite, to activate the opposite side of the balance in order to restore the homeostasis. Anyway, these rescues we saw are exciting, which could provide different strategies for treating CMT through gene therapy, including CRISPR, a field that is under rapid development in recent years. The last part of my talk today concerns another disease state linked to disruption of a tRNA synthetase regulated homeostasis. The disease is called antisensitive syndrome. I will share with you how scientists at ATAR Pharma, a San Diego-based biotech company, took their understanding of the disease into developing a first-in-class treatment for a major form of interstitial lung disease. For disclosure, I'm a scientific co-founder of ATAR Pharma. Antisensitive syndrome, or ASSD, is an autoimmune disease characterized by the presence of autoantibodies against tRNA synthetase in patients, hence the name. Autoantibodies are antibodies against ourselves. They usually do not exist in healthy people. ASSD patients have inflamed muscles, joints, and skin. Especially, lung is a commonly involved site in ASSD, manifested as interstitial lung disease. The inflammation leads to fibrosis and thickening of the lung tissue and loss of lung function, which make ASSD a potentially fatal condition. Current treatment is mostly steroid or other immunosuppressant with limited evidence for efficacy and serious side effect and toxicity when used in long term. 8 different tRNA synthetase has been described as target of ASSD, among which histidyl tRNA synthetase, or HISRS, is the most frequent target, and the web domain of the HISRS is the main antibody target site. Interestingly, HISRS was the first tRNA synthetase that acquired a web domain during evolution, and the acquisition coincided with the emergence of a primitive immune system. Scientists at ATAR Pharma proposed that HISRS, and especially its web domain, is a negative regulator of immunity and functions to suppress inflammation in healthy people when there is a environmental insult, such as infection or injury. So again, HISRS, through its new domain, is involved in this homeostatic regulation in response to the environment. However, ASSD patients, for whatever the reason, possibly some defect in their antibody response system, generated autoantibodies against the tRNA synthetase and blocked its anti-inflammatory function and lead to the widespread inflammation. Based on lots of preclinical data that support an immune inhibitory role of the web domain, ATAR initiated clinical trials on using the web domain for treating a major form of interstitial lung disease called pulmonary sarcoidosis in patients without HISRS autoantibody. Pulmonary sarcoidosis refer to small lumps of inflammatory cells called granulomas. And the presence of granulomas in the lung will eventually cause scarring and fibrosis and loss of function of the lung. So this drug is now called efzofitimod, which is the HISRS web domain fused with the Fc protein that stabilized the drug in the human body. Last September, ATAR announced a positive result from a phase 1b/2a clinical trial. It shows that the drug was not only safe and tolerable in patients with pulmonary sarcoidosis, but it also can reduce oral steroid use, reduce inflammation and fibrosis, and improve lung function. if everything goes well, this may be the first tRNA synthetase-based therapy. And the therapy is rooted in this overarching role of tRNA synthetase in keeping our body in a balanced homeostatic state in response to the changing environment by connecting the two different worlds of nature and nurture. At the end, I would like to thank all past and current members of the Xiang-Lei Yang lab. I have had the good fortune of working with many brilliant and dedicated scientists and of having the opportunity to train our next generation of scientists. The picture showing here are our current lab members, and they're from all over the world: China, Japan, Germany, France, and of course, the US. Our scientific community at Scripps has been greatly enriched with talents from home and from overseas. I also like to thank our collaborators and colleagues working in the field of tRNA synthetases, both in academia to pursue the fundamental knowledge and in the biotech industry to realize the potential of tRNA synthetases for making a difference in humanity. Last but not least, I want to thank all the financial support we have received over the years. We cannot do much without funding from NIH and the private sectors, including generous donations of individuals who understand the value of our work. I especially want to thank the Hahn family for their support and honor provided to us through the Ernest W. Hahn Endowed Chair. And thank you all for your attention.
Thanks so very much, Xiang-Lei. That was terrific, super clear. There's a lot of questions coming up. So is there a, in the first story that you told, where there's phosphorylation that keeps the synthetase away from the DNA, is there a specific kinase and phosphatase that regulate that? And could those be actual targets for maybe even small molecule therapeutics that, now that you've shown this is important, could you attack the problem in a different way?
Yeah, that's a great question and a very, very good point. So we do know the kinase that put the phosphorylation onto the cells, and it is ATM-ATR. There are two kinases involved in the DNA repair pathway. And actually this idea of targeting the DNA repair pathway to treat cancer is a very promising idea that has been having some success in the recent years. For example, the inhibitors of PARP, which is another a protein or involved in the DNA repair pathway. So yes, absolutely, that this could be another way of targeting, using small molecule of targeting the cancer as well.
Right. Now, here's another question. This is a real basic question, but I think it might have occurred to some other people. And so that has to do, you showed that you could affect blood vessel development, but does this have anything to do with any other types of blood cancers? Yes, like a leukemia? I think that's something people might be thinking about.
Yeah. Well, blood vessel growth is not important for blood cancers, right? So the blood vessel growth is usually very important for solid tumors, but it doesn't necessarily. So this particular angiogenesis aspect that may not have a direct relevance to the blood cancer, but from other works that we have done in the lab, that SARS also have other target genes and actually have other functions that inhibit cancer growth. For example, it also inhibits the Wnt signaling pathway. So I would not rule out the possibility that SARS would also affect blood cancers, but probably not in the setting of blood vessel growth inhibition.
Okay. And so what happens in sort of normal tissues if you're trying to inhibit angiogenesis and in the tumor environment?
Yeah.
Does that spill over into normal tissues, and does that make it complicated?
Yeah, I would say angiogenesis in normal tissues is usually off, right? For adult person, we don't have much angiogenesis going on unless we have a cut, we have an injury. That is when the angiogenesis are happening, and also the menstrual cycles in women. That's when angiogenesis are happening. But in general, it's a pretty silent biological process in adult body.
Okay, here's a question on everyone's mind. Are there any roles in COVID-19 for tRNA synthetases?
Well, actually, another clinical trial that ATAR has done during the pandemic was to test for this molecule I talk about. I still need to remember the name, efzofitimod. It's a new name that came up. It used to call it ATAR 1923. But anyway, so this molecule was used to in the clinical trial to see if it can help COVID-19 patients. And I would say that the result was promising. It's not very clear, but the result was promising.
Okay. So here's a question that has to do with nuclear transport of the synthetase. How does it get in? Mm-hmm. How do you specifically transport a synthetase into the nucleus?
Yeah, well, synthetase in this case is not so special compared with other proteins. Generally speaking, if you want to transport something into the nucleus, you need that signal sequence that I talk about, nuclear localization sequence. If you put a nuclear localization signal sequence into a protein that usually doesn't go to the nucleus, now you can actually make the protein go to the nucleus. So it's really a tag. You have a tag, and that tag can be recognized by a protein that is responsible for transportation.
All right. So you mentioned that in passing CRISPRs. And so is there a way to use this sort of new gene editing technology to kind of affect these alternative roles for synthetases?
Yeah, well, so CRISPR is a very powerful technique, right? And basically, it's in very rapid development in recent years. And even though there's still a lot of particular issues need to be solved, but I think the future for CRISPR is very bright. And that is a general comment. And if you think about using CRISPR can do something with high precision, right? Like a point mutation, correct a point mutation. Or it can do like a specific deletion of a certain region. So in the context of what I talked about, well, in the context of, I would say genetic disease in general, that this high precision possibility of the CRISPR can be utilized to basically just correct the genetic mistake that happens at the first place. But in the case of the CMT that I'm talking about, the issue is that you have so many genes that are linked to CMT. And on each genes that you have so many mutations that are affected. And usually for each mutation, it's only associated with like a few families, not a whole lot of patients. So using CRISPR to correct the mutation one by one would be a very difficult and onerous effort. So in this case, that if we can come up with some strategy that can, possibly in principle that affect multiple mutations, if not all mutations in the gene, that would be, theoretically speaking, a preferred method.
So you focused on 3 particular synthetases and diseases. There's dozens of synthetases in humans. So, maybe what's the range of diseases? I mean, do we know lots of other diseases? Are there many to be discovered, links? How common are these domains that have been added with new functions in that very large kingdom of proteins?
Yeah. So I would say that all the synthetases have the potential of having new functions like what we talk about here. the reason that we focus on the 3 that I talk about is not because those are the only 3 that has new function, right? I would say that much of our research is driven by this disease connection that I talk about. And in the CMT case, there are 6 genes linked. And in the ASSD case, there are 8 gene links. And there is actually a good overlap between the 2. I don't remember the specific number, but I think like 4 or 5 are overlapped. For example, GLARS and HISARS are overlapped between those 2 diseases. And you wonder why, those are more likely to be linked to disease. And one way that we think about it is the mobility of the tRNA synthetases. For the 20 tRNA synthetases, about half of them are in a large complex called multi-synthetase complex. And they are basically it's like a depot, like they're holding there. They can do their protein synthesis function, but they also need to leave the synthetase. If they do some other functions, they need to leave the synthetase, okay? And the disease that I'm talking about here, the ASSD and CMT, generally speaking, do not involve the components of the multi-synthetase complex. It's the proteins, it's the synthetases that more freely mobile, is able to go to different places more freely. So I think that could be, the differences between the 2 categories. But the other group in the multi-synthetase complex doesn't mean that they're not important. It could actually mean something the opposite, because they're so important. And therefore, whenever you have a problem happen to them, you just end up to this lethality issue. And that translates to the human population, means that this mutation cannot be passed on to the next generation. And therefore, we don't see them in the disease space.
Right. So the family of synthetases, I know something about the evolution of it. This is actually a big, long question, but maybe you could give a kind of succinct answer. I mean, are these things conserved in all organisms, and and does that conservation present any obstacles for kind of using these therapeutic strategies?
Yeah, absolutely. So they are indispensable, right, for all cells in all organisms, because they carry out this most fundamental function of protein synthesis. And it makes it difficult from the point of view of research on their new function, because a common way of understanding a gene function is just basically knock it out, means you just get rid of, remove it from the organism and to see what is the phenotype, right? And the synthetases, you cannot remove it. You remove it, you definitely just have the lethal phenotype, which doesn't at a very early stage, which won't tell you much at all. So so one way that our strategy of studying it is to going after those new domains, because those new domains, generally speaking, as you can see, as you can understand from this evolutionary point of view, that it add on later, right? So it's not critical for this protein making function that they have. So if we can just get rid of the specific domain and see what happened at the organism level, for example, in mammals, in mouse models, that is a good way for us to understand this function, this broader functional space of the tRNA synthetase.
Yeah, I'm kind of astonished that you'd have these very central proteins and they're moonlighting, doing something else.
Right.
But this seems to be not that unusual in biology. Rather than create a whole new gene, you just grab something that's there and adapt it for a new purpose. So that's pretty interesting. So people have asked this question in a variety of different ways, but how general do you think this sort of thing is in particular for autoimmune reactions and autoimmune diseases?
How general? So I can kind of answer this question and the previous comments you have together, right? So it's interesting that and I hope you can see that from the talk that I gave today. Oftentimes the function we see on the synthetase are a negative regulation function, right? So this is the way I think about it. I think as the organisms start to evolve and need to develop new function, you need positive regulators such as VEGF-A, right? If you want to develop blood vessel that is a positive protein or gene, you need to add on to the genome that you did not have before. But adding on positive regulators can give you unexpected consequences. And for the negative regulators, the organism or the nature tap into the existing resource to try to provide that break.
Right.
And that's where, you know, tRNA synthetase being around for so long. And also I think this ability of the tRNA synthetase to be connected with the small molecule and the big molecule world probably make them a good candidate to do that, right? And in the autoimmune disease space, a lot of the autoantibodies are, you know, RNA, DNA binding proteins, right? Old proteins. So yeah, I think this related to that. First of all, they are there, and secondly, that they possibly also because they have this kind of role, their connection with the DNA with RNA, and also DNA RNA is an important pattern recognition molecule for the immunity as well. So I do think there's a connection. It's not coincidental that tRNA synthetases and other RNA DNA binding molecules are, you know, target of autoantibodies.
Yep. Okay, we're coming up on our hour, and I'm mindful of the time. I want to just close by asking one question. I mean, you kind of alluded to this earlier, but how did you get interested in doing science? When did you, you know, were you you know, in the crib wanting to be a scientist, or did you come by it later in life? How did it happen?
I think it's in the gene, but I just it didn't get expressed early on. Maybe there was something inhibiting.
Right. No, okay. It was probably a synthetase.
Maybe it was synthetase.
Yeah. Very good. Okay. So thank you, Xiang-Lei. There's tons of really positive comments. It was a very clear lecture. Everybody appreciates that. And I just want to say one other thing, and that's to give an ad for the next upcoming lecture. So maybe you can see it. I think Wednesday, March 16th is our next front row. It'll be Sumit Chanda and talking about the future of pandemic preparedness. So again, it's been my pleasure to host you here on the front row, and we hope to see you again very soon. Thank you all for coming.